Hello students, this is the 12th video of molecular basis of inheritance and topic covered in this video is LAC operon. So let's start. When all genes regulate a metabolic pathway, it is called an operon. The operon concept was given by two scientists, mainly Francois Jacob and Jacques Monod. There are various types of operons. For example, LAC operon, his operon, and val operon. In this video, we will talk about LAC operon. Now let us study the genes involved in LAC operon. The first gene involved in LAC operon is regulator or inhibitor gene, which is denoted as gene I. This gene codes for repressor. The second gene involved in LAC operon is promoter gene, which is denoted by a symbol P. Third gene is operator gene denoted by O. Now, fourth gene are the three structural genes. They are Z gene, Y gene, and A gene. Z gene codes for beta galactosidase. Y gene codes for permease and A gene codes for transacetylase. Now let us look at the function of these three enzymes which are coded by Z, Y and A gene. Beta galactosidase hydrolyzes or converts lactose to glucose and galactose. Gene Y increases permeability of cell to lactose, and gene A, which codes for transacetylase, the enzyme transacetylase helps to transfer acetyl group from acetyl-CoA to beta galactosidase. Now let us look at working of LAC operon. We have to remember that lactose is the inducer of LAC operon. Now, why is lactose called inducer of LAC operon? It is because it helps in switching on and off the operon. If lactose is present in the LAC operon, it would be switched on. Whereas if lactose is absent in the LAC operon, it would be switched off. Now let us look at what happens to the LAC operon in absence of lactose, means in the absence of inducer. Now let us look at what happens to LAC operon in absence of inducer. Given below is the graphical representation of LAC operon. We know P is the promoter gene, I is the inhibitor gene, O is the operator gene, and Z, Y, A are three structural genes. Now when lactose is not present, the I gene, which is the inhibitor gene, will code for repressor mRNA. This repressor mRNA will produce repressor. This repressor will bind to the operator gene. Now, when the repressor binds to the operator gene, this will block RNA polymerase enzyme to transcribe Z, Y and A gene, because the P, that is the P gene, that is the promoter gene, will produce RNA polymerase. This RNA polymerase path would be blocked as the repressor has bound with the operator. So the RNA polymerase would not reach to Z, Y and A gene, and hence their transcription would be stopped. Now let us look at what will happen to LAC operon in presence of inducer, that is lactose. So if lactose is present, again, first of all, the inhibitor gene will produce repressor mRNA, which will form repressor. But now the lactose is present. So this lactose will bind to this repressor and inactivate the repressor. As you can see in the diagram, the yellow colored lactose has bound with the red colored repressor and it has inactivated the repressor. So now repressor has not bound to the operator. So RNA polymerase enzyme produced by gene P, that is promoter, is now free to bind with Z, Y and A gene. Hence, their transcription would take place and they would be transcribed into LAC mRNA. Hence, in the presence of lactose, as repressor is inactivated, this whole operon is switched on. So this is why lactose in LAC operon is called inducer. So if you have gained something from this video, please subscribe now to my channel Ramji Chopra, comment, like and share.